Good afternoon. Good afternoon to the audience here and to the audience online. It's my pleasure to welcome today's NITheCS Colloquium speaker, Professor Özlem Tastan Bishop from Rhodes University, and you will see that she's from the research unit in bioinformatics at Rhodes University, where you've been since 2009. So welcome to our colloquium. Professor Tastan Bishop, actually, I see from her biography, started off as a physicist with an undergraduate degree from Boğaziçi University in Turkey, and then pursued a PhD eventually at the Max Planck Institute for Molecular Genetics and the Free University in Berlin, and somehow ended up in South Africa afterwards. Today, she's telling us about drug design, a challenging and very important topic to us all. Thank you very much for addressing us this afternoon.
Thank you. Thank you very much for the kind introduction. Good afternoon, everybody. It's very nice to be here in Stellenbosch University and give this talk in this NITheCS Colloquium. I wanted to thank Francesco for inviting me here, but he's on the way to Europe currently. And all the coordinators making this trip possible. Okay, if you look at the title of my talk, it is actually inspired by a book in the 5th century BC. It's called The Art of War. Written by Sun Tzu, and I would like to start with 2 quotes from him. The first one is, if you know the enemy and know yourself, you need not fear the result of 100 battles. If you know yourself but not the enemy, for every victory gained, you will also suffer defeat, and it continues. And the second quote is, to win 100 victories in 100 battles is not the acme of skill. To subdue the enemy without fighting is the acme of the skill. So what is the battle that I would like to talk today here? The one that is actually the battle against the infectious diseases, disease-causing pathogens, and most importantly, the battle that we give continually to drug resistance. Is there even a victory without battle for the drug resistance? You might think that I'm very optimistic to think about that. But I believe that actually, if we can preempt the drug resistance issue while developing drugs, perhaps we can reach, as Sun Tzu says, acme of skills in drug research and development. But unfortunately, we are nowhere near to that, and pathogens are always a step ahead of us. I would like to share this table with you. This table shows the top 20 causes of death in African continent, which was published, taken from analytical fact sheet published in June 2023 by World Health Organization. And the red lines that you are seeing is basically shows the increase from 2000 to 2019, the cause of death increase, and the green one shows the decrease. If you just look at this table, you can see that there are actually lots of infectious diseases as the 20 among these 20 top causes of death in Africa. But this is not a surprise to us, right? Africa suffers from communicable and non-communicable diseases. We all know about that. Let's zoom into the individual countries. When we look at individual countries, we see that the scenario differs from one to other. For instance, for South Africa, top killer is HIV/AIDS, according to this report, followed by lower respiratory infections. On the other hand, country like Democratic Republic of the Congo or Angola suffer more about other infectious diseases like tuberculosis, malaria, even measles, and lower respiratory infections. So whatever we see here about infectious diseases and the drugs, I mean, we clearly have problem. And I think the challenge is twofold. So I'll try to discuss what I'm thinking about what the challenge is here in the diseases and drug discovery. I think the first challenge is itself the drug research and development. It's a complex process, but it really takes very long time. To get a drug out of this process, it takes at least 15 years, 15 to 20 years. We can broadly divide this process into two, discovery part and the development part. And most of the research groups work in the discovery part of drug R&D. And my research also falls into perhaps where it says exploratory research. Okay, but what is really important here to take note that development stage merely depends on what is coming from the discovery stage. So development stage can be further subdivided into two as preclinical and clinical, and it involves animal from perhaps part of lead optimization to animal models and then goes to phase 1, 2, 3 clinical trials. So challenge 2, in my opinion, is the high attrition rate. This figure actually very nicely summarizes the possibility of success if you follow this line here. So as you can see, according to this figure, you can start with more than 10,000 candidates. But when it comes to lead optimization, it starts, the success rate drastically drops. So obviously something is not done correctly in the discovery stage. So what happens is that it really, when it comes to the clinical stages like phase 1, phase 2, phase 3, from starting point like over 10,000 candidates, you would be lucky if you get one drug out of it. And this is, as I said, roughly here, as it's shown, I think roughly 13, 14 years. And then, of course, the problems continue for the marketed drugs because, I mean, it doesn't end there. We always face to drug resistance, no matter what. And also, depending on to what population those drugs given, we can face to adverse drug reactions. So I would like to take you to some key facts about drug resistance. This is again from World Health Organization, published in 2023. And it basically states that there is serious problem with the antimicrobial resistance AMR. For instance, bacterial AMR is directly responsible for 1.27 million global deaths in 2019 and roughly contributed to roughly 5 million deaths. So it also is very important for the medical procedures. It makes really harder to handle the surgery, CS, or cancer chemotherapy. And the World Bank estimates that AMR could result in 1 trillion US dollar additional healthcare costs by 2050. So this is actually huge amount of loss in many different ways. And we see a similar report from World Health Organization for HIV drug resistance For instance, I'm not going to go details, but remember that this is the top killer in South Africa, and we are always facing drug resistance. And of course, malaria. Even malaria is not such a problem for South Africa. We know that many other countries in Africa suffers from malaria, such as Benin and Burkina Faso. And interesting, World Health Organization has actually a published document for the strategies that should be taken care, and it has 4 pillars. And if you zoom into the fourth pillar and the third item there, it indicates that develop new treatments and diagnostics with the objective of delaying the emergence and spread resistance. How do you delay resistance? Perhaps more drastically, how can you prevent drug resistance? But our first question is here, why do we have drug resistance? I mean, it is no matter what drug we have for the pathogens, in certain point it gets drug resistance and leaves all these years of effort, millions of dollars cost in a way ineffective. And this is due to pressure. Pathogens changes in pressure, and drug treatment is a sort of pressure to them. And one of the key drug resistant mechanisms under this pressure is genetic mutations. So pathogens basically continually evolve to escape from the environmental pressure, in this case, pressure coming from the drug treatment. So that then bring me to again back to this pipeline and reiterate again to say that discovery stage of the drug development is highly important. And unfortunately, there is not much done in consideration of mutations while looking at the hit generation. So what happens normally, especially in computational drug discovery, but in any screening, the compounds are selected and then put into some kind of resistance strains and checked, but there is no actually analysis done in a way that it looks every possibility of the current mutations, how it would affect, and the future mutations, how they would cause the drug resistance. So what we propose in my group can be divided into 3 as a solution to get longer lasting, more effective drugs. First, we need to understand the underlying molecular mechanisms of the effects of missense mutations on drug target proteins at atomic level. This is really important. And there are many ways that one can do that, but I mean, there is enormous amount of data, actually, current data for the current drugs. So that should be really analyzed because that will give us information about how the pathogens are evolving to escape from the pressure. The second is also very important. We need to predict the future mutations. Can we predict for instance, and a virus, how it's gonna mutate to escape for the future future drugs, and can we design these future drugs by using this prediction? And the third important point is designing more multi-stage PAN inhibitors, but in the consideration of, again, mutations. So normally, most of the drugs are targeting just one thing, and actually, it is very easy for pathogens to evolve around that target. But if we are targeting many different drug targets at the same time with one inhibitor, then it would make their life harder. So today I will be focusing the first one, which is understanding the underlying molecular mechanisms of the effect of missense mutations on drug target proteins. And I will be giving examples to first current resistant mutations and how they actually affect the current drugs. And then I will talk a little bit about using the information from the current mutation, how we could design future drugs. Okay, so I am telling that it is really important to understand the molecular mechanisms of mutations, but this is actually not a very trivial job to do. Um, I like to show this slide. I showed in some of my other talks as well. So what we have here? Let's say a protein, and with the functional site, we call it orthosteric site. And if there is a mutation in the functional site, yes, that's easy to understand how that mutation is causing drug resistance, because probably most of the drugs are designed for the functional sites, and this mutation is probably directly affecting the binding of the drug to active site. But life is not so easy. Nature is very clever, and most of the time, mutations are far from the active sites regions. And there's a reason for that, because actually, pathogens do not want to lose their proteins, but they want to change the protein in a way that it would affect the binding of the drug, not the function of the protein. So these mutations far from the active site or functional site are called allosteric sites. Okay, and the mutations are called allosteric mutations. And sometimes number of mutations, not only one or two, many of them come together to affect the function of the protein, and they work collectively. And we actually last year published, 2 years ago now, published an article talking about these mutations and their collective effects. So I like to define allostery as action at a distance, because this is what it is. So allosteric effect, allostery is actually intrinsic feature of micromolecules, the proteins. So any protein can has this, many proteins have this feature, and many things, many different things in the cell can cause the allosteric behavior of the protein, like post-translational modifications or drug binding or RNA, DNA binding and the mutations. Okay, so these things bind far from the active site, but can cause structural and functional change in the protein. So today I will be talking about drug and ligand binding, and as well as mutations. So just to explain a little bit about allosteric communication, I like to do this analogy to Chinese whisper. You basically give an input somewhere, and all these people like the residues in the protein, and that input comes as an output somehow in the active site. So we started to look at missense mutations and their effect roughly 7 years ago. And this is our very first article, actually. It was interesting that time there was hardly any work done in understanding the effect of missense mutations in the proteins. We came up with a kind of protocol, which is really, details are not important here, but what is really important for us to focus here. So we thought that we could basically do molecular dynamics to understand the effect of mutations on the proteins on global level, but we also need something beyond that to understand how the residue level and the mutations affect the protein residue level. And we developed something called dynamic residue network analysis. So network analysis is not new. It's coming from social sciences. And actually, there were some studies then when we started doing that, but all of them mainly they were looking at static proteins. So what is the idea behind it? Basically, what we are saying, all these residues in a protein communicate with each other, they are linked to each other, they make a network. And if there's a mutation, that network might change within the protein. So, of course, this is easier to make it more accurate. Actually, we just carried this network analysis over molecular dynamics simulations. This is an example. If you don't know what molecular dynamics is, basically we put the protein or the protein complex into cell-like environment and calculate the behavior. Okay, so we have actually tools we developed the first tool, which was NITheCS, and then more recently we established web server of the tool, which you can basically calculate the networks dynamically. So what do you get out of network analysis? So you can get number of things actually. There are different metrics and measurements, and they are because it's coming from social sciences. It can be related. I'll try to explain with the relation to social science. Like between its centrality is basically identifies the residues the most frequently used in the communication's shortest path. Why this is important? Because we believe that in within a protein, actually communication goes through the shortest path, and mutations can change this path, or binding of drug can change this path, meaning that the residues, the important residues changes. And what you get here is like a control of information. You identify the residues with the controlling the information. Closeness centrality looks at the average of the shortest distance to all other nodes in a graph. This is like diffusion, rapid diffusion point. So it's like in a society, there are certain people, you give the information, that information spreads very quickly. So that's these are the residues, basically diffuses the information the quickest. And the eigen centrality basically looks at the important residues in the protein linked to functionally other important residues. So how do we use this information now? First, I would like to take you to our very one of our very early studies in HIV drug resistance. And this was done by my PhD student and collaboration with Prof. Nigel from the Math department. So what I'm showing here is basically the resistance mutations in HIV protease. It's a homodimer. And this is the combination of all the resistance mutations coming from many, many different sequences. And you can see that mutations, this is the active site here. And the mutations are, as I said, merely in the active site because the virus doesn't want to lose its protease. So the virus aim is to get rid of the drug, and the mutations are all around, except perhaps very little in the active site. So we looked at 8 HIV protease drugs, and these are the abbreviations for each of them. And our question here was: is there a common resistance mechanism? Actually, how do they? Because whatever, no matter what you design for HIV protease, you get resistance at the end. This is the experimental design. 8 of the antiretrovirals, and we gathered 100 highly resistant sequences per antiretroviral and 100 hypersusceptible sequences. So the idea was, can we just see difference between these two? And we modeled 1,600 structures, drug protein complexes, put them through MD simulations, repeated twice, and we apply some statistical analysis to show that actually our results are significant. So the outcome was very interesting, and we were not expecting anything. Actually, we had no expectations. We were just trying to understand. But what you are seeing here, basically, this is the residues going further from each other in the MD simulation, and these are the region contracting, and each box is for different antiviral drug. So there is what we are seeing here is a kind of common mechanism, right? I mean, basically, the protein is expanding laterally and shrinking from the bottom, and that is we in that time hypothesized that it is to dislocate the drug. So this is how it's handling, causing the drug resistance. And no matter what you design, obviously for the active site, you would get the same behavior. So this was what we thought about that time. So this is also very good example to collective effect of a mutation. So many of these mutations were coming together to cause the resistance. So next thing that I would like to talk is about TB drug resistance. Here we looked at pyrazinamide. It's very hard to pronounce. We call it PZA. Okay, so this was a number of my students came together to do this project. Here we looked at 83 different resistant mutations. All of them are in this protein called PZAs, and this is where the drug sits. There is iron molecule. Okay, and you can see that these spheres are for the mutations, resistant mutations, and they are colored differently because we colored them according to distance from the active site. So again, as you can see, most of the mutations are far from the protein. So how these mutations then leading to drug resistance? What was really interesting is when we do the MD simulations and looked at the interaction between the protein and drug, and we saw that here clearly we are losing the interactions in the presence of certain mutations. So that actually then we went and looked at in detail, and we saw that depending on the mutation, drug was leaving the protein, but not in the wild type. So these mutations were causing a sort of change in the active site, so the drug could leave. And again, we did our statistical analysis combined with MD and network analysis. And what we saw here, again, there's a opening here, expansion, a kind of flap opening in the presence of certain mutations. So certain mutations were causing this flap opening and a kind of shrinking in the bottom part of the cavity. Again, there was a kind of common mechanisms for certain mutation which were far from the active site. Here, my third example is about isoniazide drug resistance. This is again TB, and these drugs are highly important for the TB treatment. But we get really very important drug resistance to this. So this is actually prodrug binds to this giant protein. It has actually two monomers called protomer A and protomer B. And here we didn't look at with drug because actually it is not clear where the drug is binding. So there are some results showing that cryo-EM results showing that it's binding to this part of the cavity or the bottom part of the hem. But it doesn't matter. Actually, what we did was to model the protein mutations in the absence of the drug and check how the behavior is. So what we saw was interesting. We saw actually that because this is a dimer, first of all, we saw that in the molecular dynamics simulations, each monomer is behaving differently. What I'm showing here in the here root mean square deviation, meaning that we checked the how the protein actually behaves over the molecular dynamics simulation with respect to the start point. And what you are seeing here, green is one monomer and brown is the other one. And it is while type is the monomers are behaving more or less the same. In certain mutations, there's a drastic change in the behavior of the individual monomers. So we then looked at if heme was changing in in each monomer, and this what you are seeing here is this is the interaction frequency with of the heme with the individual residues in protomer A and protomer B just converted into the heat map, and if you subtract these values from each other you get this table, and you can see that this residue in the presence of certain mutations, and these mutations actually cause severe drug resistance, there is a huge change, and this residue is highly important in the activity of the protein. Okay, so I mean, there are lots of examples that we looked at it, but what is really take-home message from all of these things was every case is different in a way. Every protein is behaving differently in the presence of the mutation. So you cannot generalize and extrapolate from one case to other. Okay, how about looking at the future drugs? How do we design a drug that inhibits a continually evolving protein? And the best example is, of course, coming from COVID, because this is something that we all lived within, and we saw how quickly it was changing from alpha to beta to gamma. So what we did, this was very early study. I think we got the data in the 4th month of COVID, and we and published this article. It was the first, to my knowledge, first publication on looking at mutation effects in SARS-CoV-2 main protease, which is drug target, and then we published the follow-up. Okay, so this is basically the data coming from April 2020, which surely changed quite a lot since then. But we had that time 50 distinct mutant sequences, and you could see that there were only one mutations. This scenario changed later on, and the more mutations started to accumulate. And again, though, I would like to emphasize that this is also homodimer, but if you look at the mutations, again, they are all around the protein, not in the only in the active site area. And these the residues and numbers shows where the mutations are, and we decided to show with the size of the letters. If there are more than one, it shows the frequency of the mutation actually. Okay, so I'm not going to enter the details, but what I would like to show you here, what we have here is the Wuhan structure with the structure coming from Wuhan strain, the original one, and the MD simulation. Again, we look at the root mean square deviation. And you can see that very nice behavior, very compact behavior. But in the presence of some of the mutations, this structure changed drastically. So this we observed in many different ways that I'm not gonna give the details. We looked at like the functional regions again. Here the Wuhan structure, and some of the in some of the cases it became more compact, more rigid. This region that we were looking at, and in some cases it became really very flexible. And this is about the between the interprotomer distance. We thought that probably these two monomers are changing, behaving differently. And again, you can see this is the Wuhan structure, and in the presence of different mutations, the behavior changed drastically, and so on and so forth. So what we can conclude here, though, by simple computational studies, actually we can see how the mutations are affecting the protein, and that can be a kind of indication, because most of these mutations actually disappeared later on. The ones that I'm marking are the ones stay for some time, but all these mutations affecting the protein affected the virus as well, and virus decided not to go with those mutations. So we identified allosteric pocket in this protein, and from there we identified six modulators, six compounds that we thought that it would be potentially inhibiting this protein. As many other people did, we screened and we identified compounds. But what we thought, okay, so you look at Wuhan structure and you get this compounds, but actually this protein is evolving so fast. So how the mutations would affect these inhibitors that we identified? So what we did is then we basically modeled the protein with these compounds in the presence of mutations and did further MD simulations. And the results are as follows. Some of the compounds, like this one, which was pretty stable in the Wuhan structure, showed enormous fluctuations. So this was the worst compound to continue actually. And some of the compounds, like this one, didn't change its behavior. It was stable in the presence of all the mutations. So this is something that I believe one should incorporate to inhibitor design while looking at identifying new hits. So further than that, we looked at the network analysis. I'm gonna just show, we looked at 1530 systems with different network matrices, But, and this is just one example. What I'm showing here is actually, this is the compound, the least stable compound in the presence of mutations, sitting in the allosteric site, and this is the active site. And we are seeing beautiful communication path between the ligand and the active site. And that communication path is actually is broken in the presence of a mutation, and this was a common behavior for this ligand, and we saw the similar thing for the most stable ligand, but in less manners. So, as a conclusion, I would like to emphasize, I didn't type because there's too much to say, but also very little to say. I believe that if we want to really get around drug resistance, we really need to consider the mutations and incorporate the mutation analysis to the every aspect of computational drug discovery. With that, I would like to acknowledge the funders, of course, over the years, but also CHPC. Without CHPC, we couldn't do any of the molecular dynamics, NITheCS, and also the Africa Europe cluster of research excellence that I'm part of it. And I also would like to acknowledge my university and my research group. Thank you very much.
Thank you very much. So we'll open the floor to questions. Any questions you'd like to ask?
I'm just interested to find out, what do pan inhibitors do in terms of after the computational drug discovery?
Pan inhibitors idea is not very common. This is something that we started looking at it. And the idea is it's for pathogen is very easy to change one protein, like if you are targeting only HIV protease. So it is easy to change that, mutate that as I showed. But if there are like 3 proteins in HIV virus that you are targeting with one compound, then it is harder for the virus to change 3 of the proteins, mutate 3 of the proteins at the same time to get rid of the drug. So that's the idea behind it.
Okay, thank you.
Thank you. Any further questions?
Thanks for your interesting talk. I'm a little interested about, you use the network science and network medicine, if I'm not mistaken. How do you know that those two different part of protein talks to each other?
Oh yeah,
The
okay.
weighted graph.
I didn't explain the details there, but what we do is actually what you are seeing here is conversion of each residue gets a value, and then we look at like top 5% of these values, and you can do individually, or you can look at in the whole system. You extract top 5%, and then you color code, and this is what you get. So what you get is like the residues important in that metric that you are calculating per system. And then if you map these residues, which is the top 5, the most important residues. And as you can see, it's different from one system to other. So this is basically straight extracting top 5% of the residues with the highest metric values and mapping into the structure.
Thanks.
Yeah, thank you. How reliable are the molecular dynamic simulations? I mean, do you have to make a lot of approximations or do you expect the simulations to get better with time or it's sort of a question.
Yeah, no, it's a very good question. It depends. It depends how you are doing molecular dynamics simulations because it has lots of parameters, and I go through this with number of my students. First of all, it has to be reproducible. We do all atom molecular dynamics, and that is the best that we can do. So it doesn't do we don't do coarse grain, but we look at every atom. And then what is really important is actually reproducibility. And for that, because what we want to see is the change in molecular dynamics is due to mutation, not due to some system error. So what we do, we repeat the wild type calculations at least 3 times. And if we see very consistent results, then we think that our parameters are correct for that system. And then we start applying to the mutants. But we have to repeat it. And the length is it really depends. I mean, if you are working in very big system, we try to go until it equilibrates, and you see with certain calculations that it is equilibrated and the system is not prone to change anymore. So you can do different things. You can do many short MDs and apply statistical models, or you can do long MDs. It really depends. But it is reliable at the end if you do the controls right.
Okay, thank you.
Thank you. Any other questions from the auditorium? If not, are there any questions online? No. Can I just ask one question? So if you're thinking about the folded structures of these proteins, do you use crystallographic data or AlphaFold data or not?
Yeah, AlphaFold it is. If we can find crystallographic data, yes, we do. We do use it, and especially if it's a good quality. Not every crystal structure has good quality or correct structure actually. If necessary, we do modeling and we do look at AlphaFold. I did some modeling myself recently, and what I saw is actually AlphaFold can be very useful to fill the gaps between homology modeling and crystallographic structures. But all alone, it doesn't have always the correct answer, actually. It really depends on the protein. Yeah.
Thank you very much. If there are no further questions, let's thank our speaker again.